Hey guys, welcome to this video. In this video, we will be discussing how microbiome affects stress-related and neurodevelopmental disorders.
The term microbiome refers to the collection of microbes, such as bacteria, viruses, and fungi, which can be found in a single location. These microbes often have a symbiotic relationship with the host and can provide important functions such as breaking down crucial compounds that the host was not able to achieve otherwise. However, there can also be negative effects depending on what makes up the microbiome. There are trillions of microbes, all from a wide range of species and with a great genetic variation as a whole. Because of this, it is difficult to determine exactly what aspects of the microbiome cause what effects. The human body has many areas that host microbiota, such as the skin, gut, oral cavity, and lungs, to name a few. The gut in particular is widely studied, in part for its interesting links to neurological problems such as autism, anxiety, and depression.
Preclinical studies have demonstrated the prominent role of the microbiota in the gut-brain interaction. This is known as the gut-brain axis. A bidirectional relationship is found between the gut microbiota and the brain. The microbiota sends biochemical signals to the brain which influences on neurotransmitters, stress, anxiety, and behavior. The brain influences microbial balance in the gut.
These anxiety and stress-related behaviours depend heavily on functional communication between two regions of the brain, the amygdala and the prefrontal cortex. The amygdala is involved in processing and expression of anxiety, stress, and fear-related signals. Current research focuses on assessing changes in neuronal transcription in the amygdala of germ-free mice. The prefrontal cortex facilitates memory storage, behavioural flexibility, and attention. It is linked to a number of neuropsychiatric disorders, including depression, schizophrenia, and autism.
Bred in sterile conditions, germ-free mice lack the normal flora found in normal mice. In a study, when stress hormone output was measured, it was found that germ-free mice had hormone levels higher than normal control mice that were exposed to the same restraint. However, scientists found that they could induce more normal behavioral responses simply by pre-treating the animals with a single microbe. When one strain of germ-free mice was colonized with gut bacteria taken from another mouse strain, the recipient animals took on aspects of the donor's personality. Naturally timid mice would become more exploratory, whereas more daring mice would become more apprehensive and shy. These tendencies suggested that microbial interactions with the brain could induce anxiety and mood disorders. Neurostudies are showing microbiome imbalances could play a role in autism. Epidemiological data showing that women who suffer from high, prolonged fever during pregnancy are up to 7 times more likely to bear a child with autism. These data suggest an alternative cause for autism besides genetics. To investigate, researchers injected pregnant mice with an immunostimulant that induced flu-like symptoms. The F1 generation displayed all 3 core features of human autism: limited social interactions, a tendency toward repetitive behavior, and reduced communication. In addition, the mice had leaky intestines, which was important because anywhere from 40 to 90% of all children with autism suffer from gastrointestinal symptoms.
Future research. Analysis into genetic capacity of the gut bacteria in proteomics will provide essential mechanistic information for future research into novel therapeutic applications such as probiotics, antibiotics, and dietary manipulation. To further research the effect of probiotics and antibiotics on those affected by neurodevelopmental disorders, various tests on germ-free mice can be performed by inoculating the mice at an early age with specific microbes such as Bacteroid fragilis. This is a way to study the microbe's effect on the negative behaviors associated with neurodevelopmental disorders. This can be used to study the effect of fecal microbiotic transplants as well as antibiotics to combat unregulated microbiomes. As children with neurodevelopmental disorders tolerate only a narrow range of foods within their diet, producing specialized diets with specific changes to nutrient content can cause alterations to location and composition of the gut microbiome to influence the serum metabolic levels. Diets that increase levels of omega-3, gluten-free, casein-free, and low carbohydrate diets, such as the ketogenic diet, can result in reductions in total gut microbiome counts, which may increase sociability, decrease repetitive behavior, and improve social communications.